Dear children, I'm here to help you with solving a sample paper in biology. So randomly I've taken a sample paper. Let's try to solve this paper, fine. So section A. So how I'm going to help you is like to solve the paper with the help of your textbook. So you know very well, as far as CBSE is concerned, textbook, going through the textbook is mandatory. So you'll be getting all questions. Majority of the questions, I could say you 99% of the questions you get from your textbook. So if you go through your textbook, for sure you can score high marks in biology, and that will help you in your NEET examinations too. So let's learn with the textbook. So I recommend you to have your textbook with you while learning how to solve this paper. Please have your textbooks with you, children. Now the first question. From a sewage treatment plant, 3 water samples A, B and C are tested for BOD value, and the recorded values of BOD are 6 mg per liter, 400 mg per liter and 20 mg per liter respectively. What is the correct about this sample? So as I've mentioned here, this question is from Microbes and Human Welfare, and I've mentioned the page number of the rationalized textbook. So let's see the part from the textbook. Now this is a part, Microbes and Human Welfare. Please take this part, primary treatment, and here secondary treatment or biological treatment. You can see the word biological or biochemical oxygen demand. Okay, so what is this oxygen demand? So once the oxygen demand of the effluent after the secondary treatment, if the oxygen demand of the effluent is high, then it means there are a lot of pollutants in it. You can see the sentence given here. See the sentence given here. While growing, these microbes consumes a major part of the organic matter in the effluent. This significantly reduces. So once the microbes consume it, the oxygen demand reduces. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water was oxidized by bacteria. Okay, so it is oxidized by bacteria and is the demand of oxygen. The sewage water is treated till the demand is reduced. So if there is more and more demand, more and more pollutants are there to get polluted. So that's why the demand is high. Okay, so BOD test measures, so it is clearly mentioned here that BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus directly BOD is a measure of organic matter present in water. Got it? So greater the demand, more it is polluting potential. The pollutants are more. So that's why the demand of oxygen is more. If the demand is less or reduced, the pollutants are less. Okay. Now let's see the question. So here they have given 3 samples, sample A, B and C. So sample A, the BOD value, that's what they have written here. BOD value is 6 mg, 400 mg per liter and 20 mg per liter. So where the BOD value is more? In B, the BOD value is more. And A has got the least BOD value. And C, it is less actually. It has got reduced. Now, what is correct, or among the 4 statements given, which is the correct statement about these samples? Let's say the first statement. Sample A is taken from the untreated sewage. No, they have treated it well. That's why the BOD has reduced. So, less BOD, less pollutants. Keep that in your mind. Sample B belongs to secondary effluent of sewage treatment plant. No, because comparatively, sample B has more BOD. The BOD value is high. So it is not the secondary effluent. It may be the primary effluent. Now, sample C is taken from the primary effluent. No, comparatively it is less. Sample B is collected from the untreated sewage. Yeah, that's right. Because there the BOD value is high. So the answer is sample B. Got it? So this is how you have to answer this question. And I hope you understood how this part is important. How this area is important and what is biochemical oxygen demand. I hope you understood what is it. Clear, children? Now let's move on to the next question. Food chain in which microorganisms break down the dead organic matter. So the same statement is given in your textbook in the chapter ecosystem. So let's go on to the chapter ecosystem. Please take the chapter. Now we have taken the chapter ecosystem. So let's see where that comes. In ecosystem, it comes here. Can you see this? statement given, detritus food chain begins with dead organic matter. Do you see that? So detritus food chain has dead organic matter. It begins with the dead organic matter. Clear? And it is made up of decomposers, which are heterotrophic organisms, mainly fungi and bacteria. Now, so that's why their nutrient requirements are met by degrading the dead organic matter. The dead organic matter, which is otherwise called as detritus. Now let's check the question. The food chain in which microorganisms break the dead organic matter? Yes, of course, it is detritus food chain. Clear? Now, which of the following contraceptive methods involve a role of hormone? So you can see here a mixed up of different contraceptive methods. Here they've talked about the pills, emergency contraceptives, and barrier methods. No, barrier methods usually don't have the hormones, right? Barrier method, lactational amenorrhea pills. Lactational amenorrhea and barrier method don't have the hormones, but pills have. LNG20 pills, emergency contraceptives, copper T pills, emergency contraceptives. So this both we have a confusion. So let's check the chapter reproductive health. Please take the chapter reproductive health. Let's check the contraceptive measures, contraceptions. Yeah, it comes here. So here are different methods. Condom, the barrier method they have mentioned. Then intrauterine devices like Lippes loops, CUT, copper releasing IUDs, hormone releasing IUDs. So hormone releasing IUD, you can see one of the hormone releasing thing, it's LNG20 they have given, right? Progestasert and LNG20. And then hormone releasing IUDs. Then in addition to that, then you have here oral administration, small doses of progesterone. So they are again pills in the form of tablets that regulates or that changes our hormonal secretion or hormonal balance is being managed. And they have also mentioned here, like see, administration of progesterones or progesterone estrogen combination, right? Or IUDs within 72 hours of coitus have been found to be very effective as emergency contraceptives. So emergency contraceptives also we use hormonal managing medicines. So the answer of the question shall be LNG20 pills and emergency contraceptives. So this is the answer. Clear? So this is how you have to think. You cannot like learn a part and directly answer that question. No, that kind of things are not possible in CBSE. You have to learn the whole chapter. You must be able to analyze it and then answer. Analyzing skills are very, very important here. Now coming to the next one. MALT is muscle associated lymphoid tissue, mucosal and lymphoid tissue, mucosal associated lymphoid tissue, memory associated lymphoid tissue. So you know that is something related to lymphoid tissue coming out of the chapter human health. So what is the answer? I hope most of you have got the answer, but still we'll confirm the answer. Let's take the chapter human health. So here we have the chapter human health and diseases, and you know where does it comes? It comes under, yes, lymphocytes here, right? Can you see here? Mucosa associated lymphoid tissue, MALT, mucosa associated lymphoid tissue. Now what's answer? Yes, mucosal associated lymphoid tissue is the answer. So anytime, whenever you learn to try to solve a sample paper, please have your textbook with you. If you get some doubts or slight doubts, just clarify it at the moment. Underline that part in your textbook. So the next time when you go through the textbook, it will help you a lot. Clear? Next question. Evolutionary convergence is characterized by, so convergent evolution, okay? So development of dissimilar characteristics in close related groups, development of a common set of characters in a group of different ancestry, development of characters by random mating, or replacement of common characteristics in different groups. Bit confusing? Let us get into the chapter. Chapter evolution. Now, chapter evolution. Hope you all have taken the textbook. You can see the convergent evolution here. So, what is convergent evolution? So, what you see here, it is actually what you see in this figure. It's adaptive radiations of marsupials of Australia, that it is diverging to different areas, right? Now, here in this, you can see it is convergent evolution. That is, different mammalian placental mammals, they have converged to one area and they've got an adaptation of being marsupials, right? So we can consider in this evolution in that aspect, that is, they are converging. Different placental mammals living in different area have been converged to an area and they have adapted to that area being marsupials. So that is called as the convergent evolution. Now let's check the question. The question they are asking about the evolutionary convergence. It is characterized by, let's see the options, development of dissimilar characteristics in a closely related. No, here we are talking about convergence. No. So closely related groups, we should go for more similar characteristics, right? Now, next one is development of a common set of characteristics in group of different ancestry. Yeah, that states right, isn't it? Development of common set of characteristics in a group of different ancestry here. The common set of characteristic is being marsupials. Then, so let's check the other options also. Development of characteristics by random mating. No. Replacement of common characteristics in different groups. No. So comparatively, this option seems to be the best. Development of a common set of characteristics in a group of different ancestry. From different ancestry, they have come together and they have got a common set of characteristics. So that kind of evolution, we wherever we see, we call it as a convergent evolution. Okay. Now, next question. In a 3.2 kilo base pair long piece of DNA, 820 adenine bases were found. What would be the number of cytosine bases? Yes. Which chapter? So remember which chapter it is based on? Yes. Molecular basis of inheritance. Okay. Let's see the chapter. Molecular basis of inheritance. Here in this molecular basis of inheritance, under the heading structure of polynucleotide chain, you can see there's only one statement about that. It is given here, like based on the observation of Erwin Chargaff, that here you can see based on the observation of Erwin Chargaff, that for a double-stranded DNA, the ratios between adenine and thymine, guanine and cytosine are constant and equals 1. So let's see how shall we work out this question based on the statement. So according to Erwin Chargaff, he says that adenine plus thymine is equal to guanine plus cytosine. So let's see the problem. So let's see that adenine, that is A plus T is equal to G plus C. A plus T is equal to G plus C. And now here in this question, they have clearly mentioned that 3.2 kilo base pairs. So if it is 3.2 kilo base pair, it should be 3200. That is 1 kilo base pair is equal to 1000 base pairs. 1 kbp is equal to 1000 base pairs, right? So in that aspect, in that regard, 3.2 kbp will be is equal to 3200 bps, right? Base pairs. Now, so adenine plus thymine is equal to guanine plus cytosine. This ratio is 1. That's what meant by Erwin Chargaff. So you can rewrite this in another form, like adenine. Number of adenine will always be equal to number of thymine, and number of guanine will always be equal to cytosine. Okay. So now let's write like this. According to the question, all the 4 base pairs together, adenine, thymine, guanine and cytosine together form 3200 base pairs. And we know that adenine is equal to thymine. So what is the number of adenine bases given? 820. So 820 plus thymine 820. And G and C, it will always be equal. So you can take it X plus X. 3200. So you get 1640 plus 2x is equal to 3200. And you will get the value of x as 780. You can calculate it. You can calculate in a different way also. So I've opted this method. So the answer is 780. So the answer is 780. Clear? Given below are 4 methods and their modes of action in achieving contraception. Select their correct matching from other options, from the 4 options that follow. Okay, so let's see. Pill. Which one? Prevents sperms reaching cervix, prevents implantation, prevents ovulation. Yeah, prevents ovulation. Condom. Prevents sperm reaching cervix. Vasectomy. Yeah, sperms contains no sperms. And copper T prevents implantation. So this is how it comes. Pill prevents ovulation. Condom prevents sperm reaching the cervix. Vasectomy, semen contains no sperms. And copper T prevents implantation. So what is the option here? A3, B1, C4 and D2. Yes, this is the option. Now, competition for light, nutrients and space is most severe between obviously closely related organisms growing in the same area or niche. Obviously, competition will be more among them, between them. Next question. Which of the following immune system components do not correctly match with its respective role? So, human health. Let's see. Interferon secreted by virus-infected cells and protect non-infected cells from further viral infection. You have a doubt in this? Let's see the chapter. So we have taken human health and diseases. See, whenever we solve a sample paper and we find a question related to a certain part in our textbook, please go through the other parts, the part above and part below, that is head and tail of that part, so that it may help you to learn a certain section and so that it will reduce your burden. See? Now, the first sentence given there was about the cytokinins. You have seen that virus-infected cells secrete proteins called interferons, which protect non-infected cells from further viral infections. You understood about cytokinin barriers. So now we are learning about cytokinin barriers through 2. And cytokinin barriers are one of the example of that are interferons. These are produced by certain virus-infected cells, and this prevent the other non-infected cells from further viral infection, getting viral infection. Clear? So what was the question here? Which of the following immune system components does not correctly match? So first one, interferon secreted by the virus-infected cells and protect non-infected cells from further viral infection. So it is matching. So this first statement is correct. We have to find the incorrect statement. Second one is about B lymphocytes. B lymphocytes produces antibodies in response to pathogen. Let's check if that's right. B lymphocytes produce antibodies. Yes. B lymphocytes produce an army of proteins in response to pathogens. Do you see that? And those proteins are called as antibodies. So B lymphocytes produce antibodies, whereas T cells themselves do not secrete antibodies but help B cells to produce them. Remember that line too? Will help you later. And so the second statement is also right. Next one, macrophages. They are mucus secreting cells that trap microbes entering the body. Macrophages are not mucus secreting cells. They devour the other cells. The macrophages devour. See, as well as the macrophages in tissues can phagocytose and destroy microbes. So they don't secrete mucus, they phagocyte. They do devour them. So that seems to be the wrong statement. And next one, let's see the next one too. IgA present in colostrum in early days of lactation to protect infant from diseases. You can see. The yellowish fluid colostrum secreted by mother during initial days of lactation has abundant antibodies IgA. So what is the antibody found in colostrum? IgA. Yeah, you have the other antibodies too, which you are learning here. You are coming across IgA, IgM, IgE and IgG. Among that, IgA is found in the colostrum, the first yellow fluid. Okay, remember that. So which is the wrong statement? Yes, C is the wrong statement. Correct. Next question. Identify A, B, and C in the flowchart given below that represents the process of recombinant DNA technology. So the same figure which is given in your textbook is given here. So I have taken that figure here. So let's compare that figure. Foreign DNA, foreign DNA and vector DNA. So what we are doing first, what's the first step? We do cut it. So it's a restriction enzyme. So I shall just maximize it. It will be visible. Yeah. So let's compare. Same restriction enzyme is cutting both foreign DNA and vector DNA at specific sites. So what is A? What does A stand for? The same restriction enzyme. So, and remember, what is restriction? Is it restriction endonuclease or restriction exonuclease? They've given here two, restriction exonuclease and restriction endonuclease. So there's confusion in it. Restriction endonuclease is the one which cuts the DNA at specific site, whereas restriction endonuclease cuts the DNA at its ends. On the end of DNA they cut. So always endonuclease we use here for making the recombinant DNA. Now, what does B stand for? B, joins foreign DNA to plasmid. What is B? Ligases. Yeah, B, DNA ligase. So, we are getting our second statement right. And let's see if C is transformation. Yes, C is transformation. So, this statement B is correct. Don't go with statement A. It is exonuclease. So, exonuclease is different from endonuclease, remember. So, coming to the next question. Which of the following is not an example of in situ conservation? Yes, biodiversity and its conservation. You have ex situ conservation and in situ conservation. You have to check the examples given there. Let's get into the chapter. We are here with the chapter. So, ex situ and in situ conservation. How do we conserve? Now, what are the examples of ex situ conservation? Or in situ, let's check. With the examples of in situ conservation, legally protected as biosphere reserves, national parks, sanctuaries, sacred groves. Whereas ex situ, zoological parks, botanical gardens, wildlife safari parks. Let's see the question. They have asked for in situ conservation. So in situ conservation, biosphere reserves, wildlife sanctuaries, national parks, yes, and zoological parks comes under ex situ, right? So which is not the correct option. Children mostly, I have found that children gets confused with this. They never read the question properly. They go for like it is an example or it is not an example, or it's a correct statement or incorrect statement. First you peacefully, patiently read the question. Don't panic. Okay. So here, which of the following is not an example? Clear? So zoological park is not an example. So the right option is D. So I have ticked others to make sure that they are the examples of in situ. Got it. Now, next one. The age structure of a population influences population growth. Because age structure of a population. See, I have put the figure here. Let's check the figure. Its age structure comes like this. Pre-reproductive age, reproductive age. So these are the age pyramids, okay? These are the age pyramids. And pre-reproductive age, reproductive age, and post-reproductive age. So if the pre-reproductive age is, so you can see this is expanding. Pre-reproductive age is more. So there is a chance for increase in the population, okay? Expanding population, there's a chance. See, if the pre-reproductive state is less, see, this is a declining pyramid because the people who are about to reproduce, the number is less. Post-reproductive or reproductive phase is more. So there is a chance that they will be entering the post-reproductive stage very soon. So that pyramid will be declining. Okay. So now let's see the question. The age structure of a population influences population growth. Yes. And let's see. What is the reason? Younger females have more offsprings than older females. Not like that. Different age groups have different reproductive capabilities. Yes, different age group have different reproductive capabilities. More the number of immature individuals, lower the growth of population. No. Shorter generation time results in slower population growth. That's also no. So this is the right one. different age groups have different reproductive capabilities. So this is based on the expanding age pyramid. Clear? So this question is important actually, age pyramid. They'll be asking you different ways. Now let's come to the assertion reasoning part. Human insulin can be produced into bacterial cells using biotechnology. Isn't that right? Yes. To produce human insulin, A, B, and C polypeptides of human insulin are produced separately in bacterial cells, extracted and combined by creating disulfide bonds. It's true. Yeah. So both A and R are true, and R is the correct explanation of A. Linked genes do not show dihybrid ratio of 9:3:1. Yes, they do not show this dihybrid ratio. Linked genes. They show a different ratio. Because linked genes do not undergo independent assortment. Yes, they do not undergo independent assortment. Okay, yes, they do not undergo. So both A and R are true, and R is the correct explanation here. They do not undergo independent assortment because when they are linked, when they are closely arranged, the chances of independent assortment is less. When they are placed in distant locus in a chromosome, the chances for independent assortment is more. Now, next question. Mycorrhizae are kind of association between plant roots and fungus. Their major role is to enhance nutrient and water uptake by the host plant by exploiting larger volume of soil than roots alone can do. Right. Mycorrhizae come in a number of forms, depend on several factors such as host plant distribution, climate, soil condition, etc. Study the given figure showing plant growth with and without mycorrhizae. So you can see the plant growth without mycorrhizae and plant with mycorrhizae, and it's obvious that the plant with mycorrhizae is growing lush. Now let's see. Mycorrhizae represent a mutually beneficial interspecific interaction of fungi with roots of higher plants. Yes. In a mutualistic relationship, both organisms enter into some sort of physical exchange. That's not always true. In a mutualistic relationship, both organisms, they enter into some sort of physiological exchange, yes, but it's not always true. It's not like they do exchange and they get a mutual relationship. So I could say that both A and R are true, but R is not the correct explanation of A. Okay, it's not the correct explanation of A. It's true, but not always. Now, X-albuminous seeds do not possess any residual endosperm as it is completely consumed during embryo development. X-albuminous seeds do not possess any residual endosperm. Yeah, that's right. They do not possess any residual endosperm as it is completely consumed. Wheat, castor, pea and groundnut are all examples of X-albuminous seeds. Is this statement true? Let's check the textbook. So it comes in the chapter reproduction in plants, sexual reproduction in flowering plants. Let's come to this albuminous part. Now, the cotyledons of the embryo are simple structures. Mature seeds may be albuminous or X-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed. Example, pea, groundnut. Albuminous seeds retain a part of endosperm. So, example of albuminous seeds are wheat, maize, barley, castor. Let's see the question. They have given wheat, castor, pea, groundnut are all examples of X-albuminous seeds. Are they the examples of X-albuminous seeds? No, they are albuminous seeds. So, A is true, R is false. Simple. Coming to section B. You know which diagram it is? So state the hypothesis which S.L. Miller tried to prove in the laboratory with the help of the setup given above, and then name the chemicals found in the samples drawn from X. So what are the chemicals drawn from the samples? So they have, after this process, they have collected the samples and they examined, and then what's the results? That's what they have asked. Don't go for the raw materials used or the gases used there, okay? They are asking for the products. Let's see the chapter evolution. Let's check the Miller's experiment. So state the hypothesis which S.L. Miller tried to prove in the laboratory with the help of the setup given above. So the setup was this, right? So what did he try to prove? Life would have, that's the first form of life, could have come from pre-existing non-living organic molecules. So that is a statement given by operin, and haldane. So Miller tried to prove that. That is, first form of life could have come from pre-existing non-living organic molecules such as RNA, protein, etc. And the formation of light was preceded by chemical evolution. So this is what he tried to prove. That is, formation of diverse organic molecules from inorganic constituents. So that is what proved by Miller tried to prove that. Okay, chemical evolution. So after his experiment, this conjectured story of chemical evolution was more or less accepted actually. Now, name the chemicals found in the samples drawn from X. This one. So here in this figure, they have given the gases. No, they are not not expecting you to write the gases. Chemicals found in the samples drawn from X. That is, after the chemical reaction, what samples did we get? What samples did we get from that? What were the samples obtained? Yes. Here, I can see he created electric discharge in a conical flask containing methane, hydrogen, ammonia and water vapor. He observed the formation of amino acids. So amino acids were obtained. In one of the sample question paper, remember children, listen, in one of the sample question paper, they have asked about the related experiments, similar experiments. In similar experiments, they got sugars, nitrogenous bases, pigments and fats. Okay, so the experiment in this setup, what Miller got after as a byproduct, as a product of this experiment was amino acids. Traces of amino acids were found. Clear? We'll move on to the next question. Write a short note on hydrophily. You know what is hydrophily? You remember this word? Actually, this term, hydrophily, is not given in your textbook, but CBSE wants you to know about those terms. Okay. So sexual reproduction in flowering plants. We'll take that chapter. Sexual reproduction in flowering plants. Yeah, here. Sexual reproduction in flowering plants. Hydrophily, it is nothing but pollination. Okay. So let's take the part pollination. Here comes pollination. Yes. In this pollination by wind, anemophily. Wind pollination, anemophily. Then coming to water pollination. So water pollinated flowers are called as hydrophilous. So water pollination is what he meant. Clear, children? So hydrophily, you have to write here. So write a short note on hydrophily. You have to write what it is. Pollination by water is hydrophily. Then that happens mostly in monocotyledons. Here, water is a regular mode of transport for male gametes among the lower plants. In Vallisneria, the female flower reaches the surface of water by long stalk, and the male flowers or pollen grains are released onto the surface of the water. They are carried by water current. So you can write about Vallisneria. Then you can write about this one. In another group of water pollinated plants such as sea grasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside water. Some of them reach stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a mucilaginous coating. So this part you have to write. Is it clear, children? Yes. We will move on to the next question. Now, next question. State the role of UV light and ethidium bromide during gel electrophoresis of DNA fragments. Yeah, let's go to the chapter Biotechnology. Biotechnology, principles and processes. So, use of UV rays and ethidium bromide in gel electrophoresis. State the role of UV light and ethidium bromide during gel electrophoresis of DNA fragments. You see here, this part is very important. So in this gel electrophoresis, we are able to see the fragments of DNA only if it is, see, it's mentioned here, the separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by the exposure to UV radiation. You cannot see pure DNA fragments in the visible light and without staining. So the purpose is clear, right? So you can see the bright orange color bands of DNA in ethidium bromide stained gel exposed to UV light. So only this both will serve the purpose. If not, we cannot view it. Next question. Different species belong to genus Trichoderma are used to humans as well as to plants. Justify their roles by giving one instance of each. It means they are asking to say about the use of trichoderma, both humans as well as plants. So let's see the chapter Microbes in Human Welfare. Microbes in Human Welfare. Bioactive molecule. It's used as an immunosuppressive agent produced by the fungus Trichoderma polysporum. So this is the purpose of trichoderma, fungus trichoderma in human. Okay, it is used as an immunosuppressive agent during organ transplantation. So the patients who have underwent organ transplantation, they are given with Trichoderma polysporum, that is cyclosporin extracted from the fungus Trichoderma polysporum, which act as an immunosuppressive agent. Now let's go for the plant purpose. See, Trichoderma species. A biocontrol, Trichoderma can also be used as a biocontrol agent. It is used in the treatment of plant diseases. Okay, so Trichoderma species are free-living fungi. They are very common in root ecosystem. They are very effective biocontrol agents of several plant pathogens. You remember what I said about the analysis chapter, same chapter, Trichoderma in two different areas they have given. They have combined that and put in a question. So whenever you learn, relate it and learn. Now we have learnt about two uses or two different areas of Trichoderma. Trichoderma use, one species of Trichoderma used in the plant varieties, like to control the plant diseases, and one variety is used in human beings. Now let's go to the next question. Study the table given below in regard to population interaction and answer the questions that follow. So the same table is given. So I have taken the table, so let's maximize it. Species A, what they have given minus means they have impact. See, they have given the key here. Minus means detrimental interaction. They have some harmful effects. So species A have some harmful effect. Species B is, there's no neutral interaction, there's no benefit. Beneficial is plus. Negative, minus is some harmfulness. And 0, that is, 0 is no use. Neither beneficial nor harm. It's neutral. Okay. So here, let's see this interaction, an interaction in which species A is harmed and species B has no benefit. So let's check here. It is amensalism. One is harmed and the other one has no benefit. Second, so A is amensalism. Okay. Now, for second interaction, species A is benefited and B is harmed. A is benefited and B is harmed. Here two interactions are given, either predation or parasitism. Either of it you can write, or you can write both, parasitism or predation. The next interaction is both are harmed. In both are harmed, it is competition. Yes. And both are benefited, mutualism. Now identify the interactions A to D. That's what we did now. And explain the interaction A. Interaction A is amensalism. Amensalism is an interaction where one of the species is harmed and the other one is benefited. See, regarding amensalism, there's nothing much given in your textbook. You can explain like this, as I mentioned now. In one of the, like from the table, you can mention, you can explain. That is, it is an interaction where one of the species is harmed and the other one is neither benefited nor harmed. So, what can be cited as an example for this? It is a kind of competition. It's a kind of competition where competition between a weaker species and a stronger one. The stronger one can survive where the weaker one is harmed. Weaker one cannot, if there is a competition between a stronger species and a weaker species, the stronger species will survive. So it is not getting any benefits, but it could survive, right? But the other one, it is harmed because the weaker one cannot grow well. So that example can be cited in this regard. Now coming to the next one, list any 3 methods used by ecologists to measure the population size in a habitat. Do you get where that answer is? Let's get into the chapter, organisms and population. Comes under the unit ecology, organisms and population. So the answer comes beneath the diagram of H pyramid. You can see here. Sometimes for an ecological investigation. So let's read the question once again. The question is like this. List any 3 methods used by ecologists to measure the population size in a habitat. So it is actually a difficult task to measure the population size in a habitat. It will not always work out. So let's see how the So here, in an area, if there are 200 carrot grass, that is Parthenium hysterophorus plants, but only a single huge banyan tree with a canopy, stating that population density of banyan is low to that of the carrot grass, amounts to underestimating the enormous role of banyan in that community. So we have only one banyan tree, huge number of carrot grass. So if we say that there is only one banyan tree, so the number of banyan tree or the population density of banyan tree is low when compared or less when compared to the carrot grass. But it is it's an underestimating statement, because banyan tree is huge and it is sheltering a very huge community, right? In such cases, the percent cover or biomass is more meaningful measurement of population size. So one of the measurement is by percent cover or biomass. You understand? That is one of the measurement. Then the next one. Sometimes for certain ecological investigation, there is no need to know the absolute population density. In that cases, relative densities will serve the purpose. Let's see an example. For instance, the number of fish caught trap is good enough to measure of total population density in the lake. Number of fishes caught, relatively. Okay. Then indirectly, without actually counting them or seeing them, for example, tiger census in our national parks, tiger reserves is often used by pug marks or fecal pellets. So these are some of the methods where the population density of an area or a habitat is measured. Clear? So this is the part. It seems to be not that important, but you get question from this. Clear? So get it marked in your textbook. Now let's move on to the next question. Given below is a diagram of a human ovum surrounded by few sperms. Study the diagram and answer the following question. Yeah, it's obvious, that is from the chapter Human Reproduction. Identify B and C. Shall we move on to the chapter to check the diagram? Chapter Human Reproduction. Let's look at the diagram. Identify B and C and mention the role of C. We have to check the question once again. What is B and what is C? So this is B and this is C. Yeah. So B is the cells of the corona radiata and C is the cells of zona pellucida. Okay, corona radiata and zona pellucida. So the function of corona radiata is not mentioned in your textbook, though it is nutritive in function. It is not that mentioned in your textbook. But the function of zona pellucida layer is mentioned during fertilization. So look at the sentence given here. During fertilization, a sperm comes in contact with zona pellucida layer of the ovum and induces changes in the membrane that block the entry of additional sperm. So this ensures that only one sperm can fertilize an ovum. Clear? Now mention what helps the entry of sperm into the ovum. That's the next sentence. The secretion of the acrosome helps the sperm to enter into the cytoplasm of ovum through zona pellucida and plasma membrane. Next question. Name the specific region in the female reproductive system where the event represented in the diagram takes place. So what is the event represented here? It is the process of fertilization. So what is the event represented here? The process of fertilization. Where does it occur? Where does the process of fertilization occur? It occurs in the ampullary region of fallopian tube. Ampullary region of fallopian tube. Clear? Yeah. Next question. In pea plants, the color of the flower is either violet or white, whereas human skin color shows many gradations. Explain giving reasons. How is it possible? So they talk about the polygenic inheritance. So let's see where it comes in. The chapter Principles of Inheritance. Chapter Principles of Inheritance and Variation. Let's look for the polygenic inheritance. That's how our skin color is inherited. It's over here. Polygenic inheritance. Remember children, I have taken the rationalized copy of the textbook, new copy. So some of you will be having the old copies. Please see to it accordingly. Okay. Now in pea plants, the color of the flower either violet or white, whereas human skin color shows many gradations. Explain giving reasons. How is this possible? So here it comes. So in humans, we just don't have tall or short or like, so whenever you write the answer, you should write according to what they've asked. Okay, here they've asked about the violet or white, like they have compared the violet or white color of plants and the different color gradations in human beings. So you have to answer accordingly. So we just don't have two different. So the answer comes here. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Okay, now you have to explain that. Human skin color is a classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. The effect of each allele is additive. Then you have to explain the whole part. Is it clear? That is, A, B and C are the dominant forms of this genes. A, B and C are responsible, and recessive, small a, lowercase a, b and c are for light skin color. So once when this dominant alleles are getting added up, for example, if it's only one dominant and rest all recessive, then it is for light colored skin. It's totally recessive, then it be an albino color thing. Then as the dominant characters gets added up, we'll have the darker colors. So totally dominant, the genotype with all the dominant genes will have the darkest skin colors. Clear? Okay. And according to the number of dominant and the recessive alleles, the skin color grades may change. Let's move to the next question. Given below is one of the strands of a DNA segment. So they have given a 3 to 5' strand. Write its complementary strand. So how will we write its complementary strand? According to the complementary DNA strand they have asked for. So you have to write for T, the complementary one is A, and for A it is T, C, G, and G, C, A, T, G, C, A, T, G, C, A, T, G, C. And this will be the 5' end and will be the 3' end. So that is the complementary strand comes. And now what is the RNA strand form? So you have to use this as the upper one as the template, and you have to write the RNA strand. So for T, you get A, and for A you get U. Thymine is replaced by uracil in RNA, and for C again G, for G, C, T, A, U. So it goes on like that. So you'll get this RNA strand with 5' to 3' end. Okay, so that's how it comes. Now mark promoter, terminator, end, template and coding strand with the given statement. So the promoter end will be towards this side and the terminator end will be towards this side. So the promoter and the terminator end, you have to mark it in the template strand. So imagine this is a DNA double strand. This will act as a template and this will act as the coding strand. We call it as coding strand because the coded one will be similar to this, to the complementary one. So we call it as a coding strand. So this one is template. RNA strands will be forming a complementary to the coding, or sorry, the template one. And promoter will be at your left hand end, and the terminator will be at your right hand end, and this will be the structural or the transcribing part. Clear? Now coming to question number 25. Name the blank spaces A, B, C and D in the given table given below. Fungus, scientific name Cyclosporin, medical application. You remember we have discussed about the Trichoderma polysporum. Yeah, so that is a scientific name, and it's releases, or we can extract cyclosporin from that, and it's a bioactive molecule. And what is the medical application of it? It can be used as an immunosuppressive agent in organ transplant patients. Now next one is Monascus purpureus. It's also given here. Monascus purpureus have been commercialized as a blood cholesterol lowering agent. Okay, that is the medical application. And what is the organism? It is again a fungus, yeast. It's from yeast. So blood cholesterol controlling agent. And what is the product bioactive molecule? It's statin. Why are some molecules called bioactive molecules? Bio means we get it from the biological organisms, that is the living organisms, and we use to modify our metabolism. So they are active, they actively take part in our metabolism, so they are called as bioactive molecules. Give 2 examples of such molecules. Again you can write the same. Cyclosporin A and statin are the same examples. Now describe the mutual relationship between fig tree and wasp and comment on the phenomenon that operates in their relationship. So it is a direct question. The fig tree and the wasp relationship. You have to write that mutual relationship, mutualism between them. And not only the mutualism, it is unique because the fig tree, certain species of fig tree can pollinate it only with the help of wasp. So that phenomena you have to mention. Okay, next one. Explain the birth rate and death rate in population with the help of an example each. Let's go to the textbook. Chapter Organisms and Populations. Explain the birth rate and death rate in the population with the help of an example. They have asked for an example. So let's check this one here. Okay. So in a population, rates refer to the per capita birth and death. And here, the examples are given here. In a pond, there were 20 lotus and 8 new plants. So per capita, 8 new plants are added, 8 new births. So per capita is done by 8 by 20. 0.4 offspring per lotus per year. And 4 individuals of 40 fruit flies died. It's like 4 by 40. Is it clear? Now, next question. What is an age pyramid? Draw an age pyramid of expanding population. I hope we have already discussed this, but again we'll check the same chapter. It's about age pyramid. You have to write what age pyramid is. And then age pyramid of an expanding population. So age pyramid definition is given here. A population at any given time is composed of individuals of different ages. If the age distribution is plotted, then we call it as age pyramid. So here we have the pyramid expanding age growth. Okay, this one, the first one. Next question. How is Apomixis different from Parthenocarpy? You must know the difference between Apomixis and Parthenocarpy. Apomixis is we are getting a seed, like we are getting a fruit with seed without fertilization. So if there is a diploid condition, in some species, the diploid egg cell is formed without reduction division. That's how the seed is formed without fertilization. Whereas in Parthenocarpy, fruits develop without fertilization and those fruits do not possess seed. They are seedless one. So that's the difference between Apomixis and Parthenocarpy. And now you have to write any 2 different modes in which apomictic seeds are produced. You have examples given there. Let's see to the examples in the chapter reproduction in plants, sexual reproduction in plants. Sexual reproduction in flowering plants. Let's see that apomixis part. So there are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into an embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucella cells surrounding the embryo sac start dividing and protrude into the embryo sac and develop into embryos. And that condition is called as polyembryony. Clear? This is the answer for the second part of that question. What are biopatents? Mention their significance. So biopatency, it is actually, you can write this definition, it's the right granted by the government to an inventor or the one who designed it for the production of biological entities, that is genetically modified microbes, or it can be plants or animals or plant products or animal products or microbial products derived from them, or application of such organisms and products. So it owns that particular person owns it. It's right. So it cannot be used by anyone else. Its copyright cannot be taken by anyone else. Okay, so that is called as biopatency. So the significance of biopatency is that the person who developed, the developer is benefited out of this. Okay, he'll get the total grant, that nobody else can copy it without that owner's permission. Okay, he'll be granted for that. Now coming to section D, lac operon. They've given about the lac operon. Let's see the question. When will the lac operon switched on in E. coli? Okay, so let's see the lac operon part. So the first question, when will be the lac operon switched on in E. coli? You can see the statement is given. Lactose is a substrate for the enzyme and it regulates the switching on and off of the operon. Okay, so it is termed as the inducer. Second question, E. coli cells with mutated Z gene cannot grow in medium contain only lactose. So you imagine the medium is having only lactose. The lactose has induced the operon, but Z gene is not working. What's the purpose of Z gene? Z gene produces beta galactosidase. So only in the presence of beta galactosidase, lactose can be metabolized. So if there is no beta galactosidase, then there is no use of lactose. So E. coli cells cannot grow. That's why they cannot grow if the medium or the substrate contain only lactose. If any other substrate or any other sugar forms are found, they can survive. But without the presence of beta galactosidase, only the presence of lactose will not help them. Okay. What will happen if the operator gene is mutated? So imagine this operator gene. So this part is mutated. So if this part is mutated, then there is no role for repressor. Then there is no switching on and off of this operon. Operon will always be functional. If the lactose is present or lactose is absent, no issues. It will always be operational because there is no operator here. Okay. Then next part of the question, I think, how are the structural genes inactivated in lac operon in E. coli? Structural genes are inactivated. So which are the structural genes? Z, Y and A. These three are the structural genes. So how are they inactivated? They are inactivated by the action of repressor protein. So if the inducer produces the repressor protein and they bind to the operator part, then the structural gene will be inactivated. Is that clear? Yeah. Next question. Carefully observe the given structure and answer the questions that follow. So do you identify this? Yes, it is opium poppy. So let's see. Name the compound obtained from the given plant. So what is a compound obtained from the given plant? We get actually morphine. Yeah, morphine is the compound which we get. What is the chemical nature? So actually we can, for this first question, we can write this heroin. Heroin is the compound which we are getting it. That is diacetyl morphine. Since the second question they have asked about the original form, the chemical nature of the compound obtained from the plant, we can go for morphine because by acetylation of morphine, we get this diacetyl morphine or heroin. So you can write the first question, answer as heroin or diacetyl morphine, and the second one is morphine. Then, what are the major effects of the compound obtained by the given plant? Yeah, it's a depressant and it slows down our body functions. Write the chemical formula and draw the chemical structure of the compound. The chemical formula of this is C17H19 and NO3, and the chemical structure is given in your textbook. You have to draw that structure, okay? The chemical structure of it with cyclohexane ring. Next question, section E. The four limbs of man, cheetah and whale and bat have the same basic structural plan. It's nothing but homology. Okay. Such organs have similar developmental pattern and they develop in related organisms, but they do differ morphologically. Morphologically, they are different. What type of evolution is exhibited by these organisms? So let's check the chapter. Let's refer the chapter evolution. This is the question. What type of evolution is exhibited by these organisms? So this is all about homologous organs, right? So the type of evolution they exhibit is divergent evolution. Why they are homologous organs? Because they are similar in their pattern, but the function the purpose they serve is different functions. In whales it's different, bat is different, cheetah and humans or mammals, it's different. What can you infer about these structures? How do we infer these structures? You can write that they have same morphology but different functions. And they all show this homology indicates common ancestry. They all have evolved from the common ancestor. So it indicates common ancestry. The four limbs of cheetah, whale and bat are used for what? Four limbs of cheetah is used for? Yes. What they use for? Running. Then a whale. Yeah, it uses flipper. Then bats for flying. The purpose they serve is different. Give one example of similar structure of the same in that homology in plants they have asked for. So here given thorn and tendrils of bougainvillea and cucurbitae represent homology. Okay. So thorn of bougainvillea, common ancestry. Thorn of tendril of cucurbita, common ancestry, but the purpose they serve are different. So we can also say that homology is based on divergent evolution. The next question. Name the primates that lived about 15 million years ago. List their characteristic features. The primates 15 million years ago. So I have taken that clip from the chapter Origin and Evolution of Man. Primates are Dryopithecus and Ramapithecus were existing about 15 million years ago. And list their characteristic features. That's also given here. They were hairy and walked like gorillas and chimpanzees. Ramapithecus was more man-like while Dryopithecus were more ape-like. When was the first man-like animal found? It was where? It was found in Ethiopia and Tanzania. Few fossils of man-like bones have been discovered in Ethiopia and Tanzania. When did modern Homo sapiens appear on this planet? So let's see that answer in your textbook. Same chapter, evolution, given here. During Ice Age, between 75,000 to 10,000 years ago, modern Homo sapiens arose. Okay, when did modern Homo sapiens appear on this planet? Around 75,000 to 10,000 years ago. How was the desired DNA for biotechnology experiments first fragmented and later separated by gel electrophoresis? So you have to write like how it has been fragmented or been cut using restriction endonuclease, and then gel electrophoresis, the separation by gel electrophoresis method you have to write. Write the steps would you suggest to be undertaken to obtain a foreign gene product. So in that case you have to write the whole step, but not in detail. You can just mention as point by point, that is identifying our desired gene, identifying a vector, then cutting using restriction endonuclease, ligating it, then how it has been transformed into a microbe, then how do we get it, how the foreign gene product is obtained from that microbe. So all those steps you can write. Clear? Now identify the figure and label the parts. The same figure I have taken from the textbook. You know where it comes? It comes in the embryo part in sexual reproduction in flowering plants. So you have to compare the figure. B suspensor, C radical, A and that is cotyledon, and D is plumule. So it's the same figure, exactly the same labeling. So you understood how much important these figures are. Now explain the development of the given above structure from the embryo sac of a dicot flower. So the development of embryo you have to write. Development of dicot embryo, okay? Not the monocot one, dicot embryo you have to write. Now study the graph given below and answer the questions that follow. So you know what graph it is. It's mentioned, clearly mentioned, that menstrual cycle. So let's see that part. Human reproduction. Coming to menstrual cycle, the graph given here. The questions are based on that. So the first question is like name the hormones X and Y. So what are the hormones? Estrogen and progesterone. Okay, estrogen and progesterone. Identify, I think that's the next question. Yeah. Identify the ovarian phases during a menstrual cycle. So as per the figure, from 5th day to the 12th day, that's what they've asked for. It's the follicular phase. Okay, that is proliferative phase. You can see that follicle is formed, follicular phase or proliferative phase. And 14th day is actually the ovulation. Ovum is released. Then 16th to 25th day, it is actually luteal phase or the secretory phase. So this is how you have to answer this question. What's the next question? Explain the ovarian events in 1, 2 and 3. So that is in the first one. First one, it is actually the follicular phase. What is ovarian event? And second, that is the 14th day, ovulation. What is the event? And a third one, that is luteal or secretory phase. What is the ovarian event? Okay. So ovarian event. Ovarian event means the events that is happening in the ovary. That's what they mean. So during the first phase, that is 5th to 12th day. What is 5th to 12th day? Follicular phase. So in this follicular phase, the term comes here. During this phase, the primary follicles in the ovary grows to become a fully mature Graafian follicle, and simultaneously the endometrium regenerates. So that's the ovarian phase. What is the phase that ovary undergo? That's what they mean. So this is enough. Then the next one, 14th day. 14th day, what happens? The secretion of the gonadotropin, and it stimulates the secretion of estrogen. Estrogen is secreted more, and this leads to the release of ovum or ovulation. Okay, then the next one, third one, that is 16th to 25th day. So 16th to 25th day is the luteal or the secretory phase. So after this, It's followed by the luteal phase, during which the remaining part of the Graafian follicle. So the Graafian follicle has ruptured and ovum has come out. That is what we call as the ovulation. And after this, the Graafian follicle turns into corpus luteum, and it secretes a large amount of progesterone, which is essential for the maintenance of endometrium and also for the implantation. Is it clear? So that's how you have to answer this question. By that, we have come to the end of this question paper. Hope you find this useful. So we'll be coming up with more solutions for the sample papers. Thank you.